So I'm still hosting her, so I have to run around all over the place. All right, so we're going to talk about the actin cytoskeleton. That's going to be today's focus. And clearly, it's important for a lot of equally important functions like the microtubules. So, central role in having all your muscle types contract. Okay, responsible for movements across any substrate. Those microfilaments are also a highway. And what you can think about these highways, because I mentioned that last time, microtubules are like the superhighway. And then if you need to get off on a local street, that's actin. So oftentimes, if you're being propelled by a motor protein, it's got to have at least two types of motors. It's got to have a microtubule motor, kinesin or diamond, and then it also has to have a myosin isoform, because myosin is the motor that moves cargoes along the actin cytoskeleton. Okay, so actin, just like tubulin, is highly conserved. There's only 6 different isoforms. There's 4 alphas, and this is just factoids, 4 alphas that are in skeletal muscle, any kind of muscle cell, and then non-muscle cells have beta and gamma actin. So that's not where the diversity of function comes from. It's always the case that it's the proteins that bind to actin. So actin binding proteins, or you can call them MFATs, microfilament attachment proteins. Those are the things that drive the function for this group of filaments. Okay, so, and then also we can't forget about cytokinesis that happens during mitosis, usually starts in late anaphase and completes in telophase. That's actually the pinching off of the 2 daughter cells. That's just like a little belt of skeletal muscle that squeezes until those 2 membranes pop off. Okay. All right, so let's look at some of the details of G-actin structure. These first couple of slides are just to go over the functions. So this is skeletal muscle fiber, and what makes this thing run is because of the actin filaments. And so we'll get into a little of those details later. Movement of cells across the substrate is due to the protrusion and polymerization of the actin filaments in a bundle and in networks that project against a very deformable, soft plasma membrane. So that's what makes this to go forward, but also it's important to be attached to the substrate. How many of you walk without touching the ground? You can't do it, right? So you have to be touching, you have to have an anchor to throw your other foot forward, and so you have to be touching here, and so that's where the actin cytoskeleton helps grab onto the substrate so it can be propelled forward by polymerization. Then it's got to take up its feet. What does it do? It has to depolymerize the actin in the back and send it to the front. So it's this continuous process of planting, grabbing, swinging forward, protrusions, and that's how the cell's going to move. So we'll look at a little bit of that. And then, of course, where's this going? If you're going to have cytokinesis, this is this contractile ring that must form, and it's basically a mini sarcomere. The functional unit of muscle contraction is a sarcomere, and this is several sarcomeres all linked together in series. All right, so we'll look at that. Okay, so back to the details of actin. We're not going to worry about what isoform is, but the characteristic is this is beads on a string. Remember, it's got a certain look to an actin filament. So it looks like a pearl necklace, basically. And the pearls are G-actin, which stands for globular actin. Now, notice this binds a nucleotide, so it must have some sort of dynamic instability. The stability of this filament is less stable than an intermediate filament, but more stable than the least stable, which is the microtubule. So going from the least stable to most stable is microtubules. In the middle is microfilaments, which are these guys. And then the most stable are the ones that lack nucleotides. But this binds not to GTP, but to ATP and ADP. Both ADP and ATP are acceptable binding partners for this to be added to a polymer. So that's not true for tubulin. Remember, Tubulin requires that the alpha and beta subunit both contain GTP. That's not the case for this. So you might ask, well, what's the role of ATP? Well, let's get the structure down first. There's basically 4 different little domains, and they have names, 1, 2, 3, 4. But that's not the important part. This little slot right here tells you, number 1, where the ATP or ADP nucleotide is going to go. But it also is open to one end, the minus end. So the minus end of G-actin faces that little cleft, which is the binding cleft in which the nucleotide fits. The plus end is where there's no opening. Okay, you can't. If you're over there with your eye, you can't see ATP. All right, but here there's a cleft that looks right in there. That's the minus end. Okay, so just like tubulin, G-actin adds faster at the plus end than it does at the minus end. But if and only if it's ATP loaded onto this. If ATP is loaded onto the G-actin, the G-actin is going to add here, then it will add faster there than it will here. What happens if this is loaded with ADP, which is quite possible? It can add here, or it can add there at the same rate. So what ADP does is it equalizes the rate of addition at the plus end. It makes it equal to that of the minus end. So the reason you usually get rid of the ADP and bind to ATP and G-actin before it adds is to make it add faster at one end. You want that polarity because it's depending on the ability to grow in one direction. And if it can add at both ends, where's it going to go? It's going to go in 2 directions, not the direction you want it to. Okay. Now we can solve the directionality by taking a binding protein and capping this end, and we force it to grow at the positive end. We can do that. But it's easier just to load this thing with ATP, and then it will add to this end preferential. So that's the reason we do that. Okay, so how do you know which way the fiber is oriented if you're looking at it in a microscope? It has to be done at the ultrastructure level, the electron micrograph. But we use a neat trick. So this is the myosin 2 motor, molecular motor. And this is the one, all the myosin family, and you get that word down, that means, oh, it's going to bind to actin and it's going to walk along with actin. It's going to split ATP at the motor, we call this the motor domain because that's where the engine is. It's an ATPase. Okay, ATPase is loaded in these little head groups. The heavy chains have 2 domains. They have a globular domain. We talked about that like the second day of class. They have a globular domain that binds ATP, and it also binds to actin. So this is the part that actually, instead of being a head, it's actually walking with its foot. I mean, it's walking with its head. So instead of walking with its foot, it walks with its head. It also uses its head to split ATP. So that's where the engine is. And this is the part that walks along this little highway, right? Or local road, right? And then there's a fibrous domain that forms a coil-coil. So that's how the 2 heavy chains stick together. It's a coil-coil motif, which you're very familiar with, all right? And then it's got some bling. It's got 3 little pieces of jewelry around the neck. These are called light chains, all right? And a lot of them are phosphorylated, and they bind calcium, and so there's very interesting things. Those little bracelets that it's wearing around the neck, it's more like a necklace. Those little short chains or light chains are regulatory in nature. Okay? So you can actually cut this all apart if you use the right enzyme. So you throw in different kinds of enzymes and you cut the fibrous domain and you throw a different kind of enzyme in there. You can actually isolate the head domain. The reason that's important is this will now stick to an actin, F-actin, filamentous actin. Okay? It decorates it. So you have F-actin that's naked, you can't see it, or you can see it, but you can't tell where the plus and the minus end is, right? And so if you throw this in there, it makes this little decoration and it forms like an arrowhead, all right? The arrowhead, the point, so an arrowhead has 2 different sides. It's got the point and the barbs, okay? The point is that thing where the arrow is going to fly. These are considered the barbed ends. All right. The point points at the minus end of the F-actin. So when you decorate an F-actin filament with these myosin head groups, it will form this kind of, they say it's a chevron, like the gas company. It forms a chevron shape, and I've sort of deconvoluted what that is because it's kind of hard to tell that that's an arrowhead. So if you blow that up, it has this little shape to it, and this is pointing to the minus end of that filament. So that means the plus end is down here, so this is going to tend to grow in this direction. It's going to grow off the bottom of the page. Right? So you need to know that because you want to know which way things are going to be moving. Right? And the myosin, here's another interesting, not just a fun fact, it's something you should know, is when myosin walks along this fiber, which it will do when it's in this form, it always walks towards the positive end. It reaches out and grabs, and it reaches out and it grabs. And it will, if it's carrying a cargo, it must carry it towards the positive end. There are isoforms that will go in the other direction. Okay, so we learned some good things about being able to figure out what the directionality of this filament is. Okay, so again, the way you determine the function of the actin is its binding partners. Okay, so you have all kinds of different proteins that bind to F-actin, and they have different functions. So let's talk about the ones that control polymerization. Remember, you're not just going to throw this into the cytoplasm. It's actually, in the cytoplasm, there's very high actin concentration. It's 0.5 millimolar. You have that 10% of your Joe Blow cell is actin. To give you a comparison on, say, a skeletal muscle cell, there's 20,000 insulin receptors. There are 500 million actin molecules. So there's lots of them, okay? And so what drives polymerization is protein binding partners, okay? Let's say you want to depolymerize, you want to prevent or inhibit polymerization. We have an inhibitor, it's called thymosin B4, sometimes it's called beta thymosin, all right? And what this does is it binds to the minus end of the G-actin, and when it's bound to the minus end, it cannot add to the filament. So beta-thymosin slows polymerization because it sequesters G-actin and will not let it get onto F-actin. So it's a sequestration protein, all right? So, but it's also a buffer because when the concentration of actin decreases, thymosin will release some actin into the cytoplasm, and then it will jump onto the fiber. So it's really not always negative. You can think of it as a storage device for G-actin, not F-actin. Okay. All right, so what if you want to stimulate binding or polymerization? You need to use this thing, and it tells you exactly what it does. Its name of this protein is profilin, and it promotes filamentous actin polymerization. That's why it gets its name. And it does that because it binds to the positive end. Why would that be an effective thing to do if you're going to take this and stick it onto an F-actin filament? Why not bind to the negative end? Well, look at let's go back to the filament, and let's go back here. Let's see, is that a good? Yeah. Okay. So if you add to the positive end, what has to touch the next piece of actin is the negative end. The minus end must be added up here. Okay? If you cover that up with profilin, it can't add to that. So profilin holds it from the positive end so that it can lay this negative end on top of that filament. That's why profilin promotes binding. Okay? It also, when it binds to G-actin, what it does, it is an adenine nucleotide exchange factor. Remember we have GAS, guanine nucleotide exchange factor? This is an adenine nucleotide exchange factor. So it displaces any ADP and ATP is picked up. Why is that important? Remember, if this is loaded with ATP, it's going to preferentially add to the plus end. And it will add to the plus end very effectively because the minus end is uncovered. So let's go back to here. So profilin, if it's touching actin, G-actin, this must be the plus end, and there's the minus end. You want to stash that onto the plus end of a growing filament. And this does this very effectively. But it does two things. It sticks it onto the growing filament, and it causes this to be lost if it's ADP, and then it makes it pick up ATP. It is an adenine nucleotide exchange factor. Okay. We have that now? That's going to make things go faster. Okay. All right, so are there any other things that stimulate, they nucleate things? All right. This is an important protein. It's called ARP23 because it's got a couple of different components to it. And so we call it ARP/2/3. Right? What does the ARP stand for? Actin-related binding protein. Actin-related protein. So it looks like actin, it smells like actin, but it's not actin. Okay, so it's actin, ARP, actin-related protein. So what it does is it nucleates growth. So it will recruit G-actin, globular actin, and it forms the minus N. It binds to the minus N, very different from profilin. It binds to the minus N of G-actin, and it stimulates its outward growth. Okay, so it is a nucleator. It stimulates that. But it also is a little bit more special than that because it will bind to this minus N, the fiber will start growing, and then another ARP will bind to that growing fiber and cause a branch point. And the branch point is always 70 degrees with respect to the original fiber. So it goes, okay, this is going here, it moves over 70 degrees, and it starts a new fiber going out this way. What are you going to create if you do that? You can't make a bundle. See, if it just did this, it would make a bundle. But this is just one actin, it's just one F-actin. It's really skinny. So this is not a good bundling protein. This makes a network. And that network is preferentially developed at the cortex of every cell. So when the microtubules coming from the nucleus bring their cargo, whether it's a vesicle or an organelle or mitochondria, and it gets to the cortex, it encounters the local roads. And it leaves, it goes off the exit ramp on the Marcia Sharp Freeway and gets on the Slide Road. This is Slide Road. Because from Slide Road, you can go anywhere you want, right? Kind of. That's why it's so busy. All right, so a network is made so you can distribute things locally, and it organizes the cortex of the cell. Because there's a lot of trafficking going to the membrane and coming off the membrane. So we call this type of complex or arrangement of actin a network, and ARP23 generates networks. Now, the other way to stimulate filament growth is with another protein. This is the Foreman protein, and there's just like George Foreman, this has 12 sons. Okay, you know the Foreman drill? This is the Foreman family, okay? And so they stimulate bundling. Well, if you have a network stimulator, you should have a bundling stimulator, and that's what this thing does. It works from the positive end, so that makes it very different from ARP23, which starts at the negative, right? And so what it does is it gets on to the fiber and recruits, actually it recruits profilin, right? And it just shimmies up the top. And that it's just continually recruiting profilin. Profilin hands off its supply of G-actin, and it adds it to the plus end. So Foreman is there just to build a single bundle of fibers. Okay? And in fact, a lot of the Foreman isoforms have whiskers. Okay? I guess George Foreman has whiskers. Okay, so this one does too, right? And so these whiskers come off, and that is the thing that binds to profilin. It's just like it's sticking things in its mouth. It's going like this. It's reaching out, grabbing profilin, and then putting the G-actin on top of the filament. And as it does that, it just moves up. I have a video of this, but I don't think we can get to it from this messed up computer. So it just builds the filament. But it does not cause branching. So Foreman tends to, do I have a bundle here? I don't think I have a bundle. Yeah. If you want a tight parallel bundle like this, you need to use Foreman. Okay. If you want a network, you need to use ARP23. Okay. And if you have a contractile, again, anything that bundles Foreman's good. And they're different. If you want to have a contractile bundle, there's an isoform, a Foreman that does that. Okay, so in different parts of the cell, you're going to have different arrangements. So in the part that's driving movement, cell movement, you have stress fibers. Okay? That stabilizes the foothold that whatever this is, probably a fiberglass, has on its substrate. So you see these stress fibers. That stabilizes it so it can project forward, just like you, when you're walking, you need to have a planted foot, and that planted foot is a stress fiber. Okay? And they tend to be bundles. Okay? But when it moves forward, it's got to lift up the foot to keep going, and so it will depolymerize those stress fibers and send the G-actin forward to be reformulated into a cortex, which is a network. Okay? And then if you want to stick out a little finger, that's called a filipodia. So if you stick your best finger forward, we call those filipodia, that's a bundle. You just take Foreman, you go, Foreman, cut me out, and you just grow a bundle using the correct nucleating protein. You wouldn't use ARP23. That's what you use to get this. To get that, you need Foreman. Okay. All right, so the type of cross-linking proteins determine what kind of formation you're going to get. Okay. These two proteins are cross-linkers. So now we're away from nucleators now. We're away from sequestration proteins. We're going to try to form different networks and bundles. How do you form a bundle? It depends on the type of cross-linker you use. If you use a short cross-linker like this one, there's two really good poster children. That would be fimbririn, and then one that's kind of cool is called Villain. Where do you think Villain is used? What has the word villus in it? Bart Simpson, right? His hair. Okay, on an inner site. That's the reason you get those little bundles. The microvilli are formed because you don't use fimbririn, you use Villain. Well, you may use a little bit of fimbririn, but they're very short. So the shorter the length of crosslinker, the tighter the bundle you're going to get. So if you want a little Bart Simpson hairdo, you need really short crosslinkers like fimbririn and Villain. Okay. Now, this is the one that's used in skeletal muscle. All right. Notice that it's much longer. Why do you think that's the case? You have to know a little bit about muscle physiology. Can you get muscle contraction without a molecular motor? No. This is the highway. This is the little highway. It's the myosin that does the movement, right? And so you have to fit a myosin thick filament inside here so it can reach up and grab and pull, okay? And what it's going to do is, so let's say this is the positive end and that's the negative end. So this would be the edge of the contractile bundle called the sarcomere. And this thing is capped, okay? So it's stable. What normally happens at the plus end? It gets bigger, right? You don't want your muscle fiber getting bigger that way. You want it stable because myosin is going to walk and walk and walk. But guess what? The thick filament never moves. What it does is it pulls this whole actin thing forward. So it's stable, but if you walk along the highway, you actually pull the highway towards you. And so everything moves to the center of the muscle fiber. So the really important thing about this little segment is you can't get that thick filament, which is made out of myosin, unless you use the proper broad crosslinker. Is this still a bundle? Absolutely. But it is a loose bundle for a purpose. Okay. So we can get tight bundles, we can get, so here's a good example. This is one of Bart's little hairs. This is a microvilla on an inner site, and out here must be the lumen, intestinal lumen. Okay? And so you have myosins and calmodulins there, but the crosslinker is Villain and fimbririn. Okay? You want a short crosslinker. All right, any questions so far? So you have to figure out what you want, and then you choose the binding partners to create it. Okay. What if, so this is a filament dimer. It is a crosslinker. It's a dimeric crosslinker. But you can tell it's really floppy. Okay. If you have a really floppy crosslinker, and our poster child for this group is filament, okay, it creates a network. So floppy dimeric crosslinkers create networks. Where do you need a network? You need them here in the cortex. Filament, we can do things with that. Okay. And then this one is a famous one. This is in skeletal muscle. This is a really, really floppy cross-linker. It's called dystrophin. This protein is huge. It's massive. I forget, where's my notes? I have something about the size of this thing. It's one of the largest genes around. But it's, so for the moment, it's huge. Okay. And that makes it very vulnerable to mutations. The more amino acids you have, the more chance you have to have a mutation. And then, of course, the classic mutation is Duchenne's muscular dystrophy. And that's when this thing becomes mutated. So why does that give a person muscular dystrophy? Okay, the role of this is to link the actin filament used in muscle contraction to form the sarcomere in that person's muscle cell to the plasma membrane. Realize that this thing is contracting, it needs to have a stability point. And in normal people, that anchoring point is a membrane protein complex called dystroglycan. So this is dystroglycan. And then it's strengthened because the extracellular domain interacts with the cytoskeleton. I'm sorry, with the extracellular matrix proteins. So you have collagen out here. So this anchor point, this stabilizes the muscle contraction process because it's linked to the plasma membrane into the extracellular matrix. So it's nailed down to the floor. What happens if you weaken this? So if you blow this with some sort of mutation and this thing never gets secreted, how is this going to link to the anchor? It's not. And when that person's muscle contracts, it rips and their muscle dies. And so that's why people with muscular dystrophy eventually can't do anything. And it's in their heart muscle too. So they have heart problems and they can't move. And so this is the culprit. Are there other kinds of muscular dystrophy? Yeah, there are mutations in that thing, there's mutations in this thing. They all cause different kinds of muscular dystrophy. Okay, so this is a medically important cross-linking protein. All right, now you're familiar with this picture. We showed it a long time ago when we were talking about peripheral membrane proteins. Remember, there's two flavors, there's direct and indirect. Most of the actin cytoskeleton are all indirect peripheral proteins. You see the fiber here, and we'll ignore all these other things, but that's an actin filament, okay? And it's linked to the plasma membrane because of this cross-linker. It's called spectrin. Notice it tends to be floppy and it's dimeric. That tends to be a rule. If it's floppy, dimeric, cross-linker, it's going to create a network. And this network provides the membrane domains that corral proteins into a particular area. Okay. How are they linked to the plasma membrane? Well, for example, spectrin will bind to anchorin, and anchorin the word tells you what it does for a living. Anchorin binds to band 3. So notice how things are all anchored to the plasma membrane, usually to some sort of multi-pass or single-pass membrane protein. Use glycophorin as a single-pass. Okay. So that's a theme developing. You have the actin cortex, but you're always going to stabilize it by binding to the membrane, anchoring to the membrane. All right. This is a special class of we're nearing the end of the different categories of proteins. So this is tropomyosin, and this is called a side binder. It binds to the side of the filament. We've seen almost every possibility. We can have a bind to the end. Those are called capping proteins. Some of them nucleate like ARC23, but they also cap that in and prevent it from polymerizing. Capping it stabilizes. Okay, so we can bind to the end. We can branch out. We can add things to it. But what about just wrapping helically around that actin filament? This is the state of an actin filament that's in your skeletal and smooth muscle. Okay? And it is coated with this tropomyosin. And the role of the tropomyosin is to prevent actin from binding to its motor, which is myosin, unless and only if calcium is present. When calcium comes into the cytoplasm as a little puff, that means that your nervous system has said contract. And so the calcium signal is decoded by these proteins here. Okay, one of these is a calcium-dependent protein, the troponin complex. It's troponin C, by the way. That's what the C stands for. It's a calcium binder. And troponin goes, we need to contract. And so what it does is it jerks on the side binder and moves. It's like moving a quilt off of a bed. It moves the quilt off and exposes the myosin binding site on actin. And then when that happens, the myosin, which are trying to get at it, can then bind and pull. So the first step is deliver calcium. Troponin goes, there's a muscle contraction needed. It jerks on the blanket, which is strobomyosin, moves it. What's it exposing? It's exposing, I guess it's not shown there. It's exposing the place where the myosin motor protein is going to bind to actin. That's what it's looking for. But otherwise, when the blanket's in place, it's keeping those sites hidden so that you don't have illicit contraction, like happens in, uncontrolled muscle contraction, Parkinson's, okay? You can see if that's screwed up, it really causes a lot of problems. So this is highly controlled. All right, so we've talked about most types of actin binding protein. Do we have any questions so far? Yeah.
When you discussed earlier protofilm, does that bind to the monomer or the
monomer?
Yes.
Yeah, it binds to G-actin. So get that. I don't know why people get confused, but G-actin is the monomer, F-actin is the filament. You remember those two words, globular actin and filamentous, you won't get confused. Okay, we're going to go into one last class, and that's the molecular motors. So let's take a break before we do that. Okay, why don't we get started again? So we're going to talk about a really interesting type of actin binding protein, and those are the molecular motors. And basically, there's three flavors that we're going to look at, and so let's go get the generalizations out of the way first. So the myosins are the families that walk on microfilaments, the actin filaments. And we'll just make the generalization that most myosins walk to the plus end. There are isoforms that go in the opposite direction. So it is possible to have bidirectional movement along a filament. Okay? And here's another thing. It's not like the little local roads here where you can only have two cars passing. You can have lots of different motors on the same filament moving with cargoes at the same time. So you can have hundreds of myosin molecules walking along the pathway. It makes it a little easier if it's a bundle, but it's still, that actin filament you're able to move molecular motors past each other on there. And so there's lots of traffic going up and down that filament in both directions. So along microtubules, you have two choices, two families. There are isoforms in each one. The kinesins walk you towards the plus. And so remember, the way microtubules are oriented, that means you're going to be moving away from the nucleus towards the plasma membrane. If you need to traffic from the plasma membrane inward, for example, what's a case where you want to take a cargo like a vesicle and move it from the plasma membrane towards the nucleus? What would be a process that would require that type of motor?
If you are taking in the insulin receptor?
In some sort of vesicle. What's the generic term? If it's not an organelle, if it's just a vesicle and we don't know what it does or where it's going, we call it a transport vesicle. So you clathrinized it, you butted it off, right? You got the insulin receptor embedded in the membrane, you uncoat, now it's a transport vesicle. Where are you going to send it? Early in the summer. How do you get it there? Well, you take a diamond motor, which moves in the negative direction towards the nucleus, bind it to the vesicle, hitch a ride on a microtubule, and walk it until it hits the early endosome. And then it's going to fuse. So this is not random. It's very controlled. Right? So remember, going towards the nucleus, you use dynein to go away from the nucleus, use kinesins. All right, so let's focus on kinesin. It's very well understood. The structure is, I think it's a heterohexamer is the correct structure. So let's look at this. It's better than pictures, so let's look at the picture. This is a standard kinesin. Okay. All right, so we're moving, that's the plus end, we're going this direction. If you're dyning, you're going that direction. This is the cargo. So there's got to be a cargo binding domain, and there's got to be the head domain. And we always know that all these molecular motors walk with their heads. They don't really have feet. Okay? And the head domain has two functions. It's the nucleotide binding area, ATP, and it's also the microtubule binding domain. So they're both in that globular domain. Okay. And so this is what the family looks like. They have various, so this is kinesin 1 that we're talking about. It's the classic kinesin. And it structured, actually, this is a heterodimer at this structure, but there's actually 2 light chains. So it's actually a hetero tetramer, right? 1, 2, and then the 2 light chains at the C terminus. Okay. And so it's got a fibrous domain. It has a neck, it has a linker. It's called a neck linker or a linker right here. So the globular head domain that binds ATP and walks on the microtubule is there. They have this neck linker domain, and then you have a coil, and then it uncoils, and you have 2 light chains that attach to the end. And it's those light chains that carry the cargo. So the heavy chains can't touch the cargo. It's the light chain that touches the cargo. So you would attach the vesicle right here. You could attach a mitochondria. You could attach a Golgi cisterna. So you could attach all kinds of cargoes there, and then you move along the highway here, right? And so this thing has to, you got 2 feet, you got 2 heads, you got to walk, right? And so what you do, this is highly processive, so it swings 1 foot, and it swings the other, and it swings the other foot. The 2 feet, or heads, I'm sorry, it doesn't have a foot. The 2 head groups are highly coordinated. They can't just bind to things randomly. It has to put one, it's kind of like us, you have to put 1 foot in front of the other, unless you're dancing with the stars and you're weird. Okay. Well, that's a different problem. Okay, so, all right, so there's the structure, the ATP binding, and now you can see those light chains are bound to the heavy chains, and the light chains are carrying the cargo. So it's going through here, it's splitting ATP, it's converting chemical energy into mechanical energy. How does it do that? Okay, well, let's look at this, and then I'll show you, because this is static, and so it's harder to get it by looking at something that doesn't move. So, before either one of these heads are bound, they're bound to ADP. So in the unbound state, it's ADP. So what happens is, when it puts its foot forward, and I'll explain how it goes forward, it puts the foot forward onto beta tubulin. Okay. And when it touches beta tubulin, the beta tubulin acts as an adenine nucleotide exchange protein. So it says, you got ADP, get rid of it. And so the binding of that leading, and so you got to distinguish which foot is leading, which one's lagging. So the one out front is leading, the one behind is lagging. Okay? So the leading head touches down to beta tubulin. The ADP is ejected. ATP, because the cytoplasmic concentration is about 10-fold higher, jumps on preferentially. So now you're loaded with ATP in the leading head. Okay, right. So what happens is that nucleotide exchange causes a conformational change. The binding of ATP causes the neck linker to bend downwards and touch the microtubule. So it's the binding of ATP, not the splitting, that does that. So that neck region has sort of an elastic property, and it's kind of like bending your knee, okay? And that bending motion pulls the lagging foot forward. But what has to happen before you can lift your back foot, you split ATP and get rid of the phosphate and ADP. Get rid of the phosphate and hang on to the ADP. So your lagging foot is now moving forward because the neck linker is bound to the microtubule. And so now you're swinging forward with the lagging foot becoming the leading foot. What's it bound to when it's swinging in the air? ADP. Okay. Then it's going to bind. The binding will cause nucleotide exchange, lose ADP, pick up ATP. What does ATP binding do? It causes your knee to bend. All right? The neck linker bends. That causes the other foot to swing forward, but only if it's split ATP. So this has to be very coordinated, bind ATP, split ATP. But this processivity, that's what it's called, moving will happen for hundreds of nanometers because it's very tightly regulated. You know, the feet are not going backwards or forwards, they're always going one right after the other. And this takes a discrete 8 nanometer step. That's because it's 8 nanometers from that beta tubulin to that one. And so it's only touching the beta tubulin. Okay, let's put all those ideas together in this video. This has been done by a very famous cell biologist. Oops, here it is. I think you can see that. Almost better than if I blow it up, it's going to get really fuzzy. All right, so what's happening here? Both of these heads start off with 8 ADP. If it's not touching anything, it should have 8 DP, right? So the first one to touch down is going to be the leading head. The one that's coming behind it is the lagging head. So let's follow this. Normally it would narrate it in your DVD or on the website. You can get this video. Wiley Vale is the one who did this. So, all right. So now it's binding. That leading foot is binding very tightly to the beta subunit. What's going to happen next? It's going to eject ADP. It's going to pick up ATP. All right, so did you see that nucleotide exchange? That's the effect of the beta subunit on this myosin molecule. That's the leading head, the leading foot. No, it's the leading head, right? Okay. And so it's exchanged. The next motion is the knee bending. And it doesn't have a knee, it doesn't have a foot. It has a neck linker. Okay? See it come down? That little yellow thing is the neck linker bending. And so that's like a knee joint. And when it does that, that motion swings the lagging foot ahead, forward. That's now the leading head. Why could it do that? Well, this thing, in order to release from the beta subunit, must split ATP and then release the phosphate. It hangs on to the ADP, so watch that happen. Oops. That's magic. It went away. Okay. Let's watch a couple of them. So see, it fired. So now it can lift off. Exchange, neck linker, swings the foot forward, touches, loses ADP, picks up ATP. This one fires, now it's loose, puts the neck linker down, right there, swings the lagging foot, becomes the leading. So this just goes over and over and over. And you can memorize this by looking at this video and you see exactly the sequence of stuff. Right? Are there any questions about that? Yeah.
Does the neck linker region touch the albumin of
that I'm not sure about. I'm sure somebody knows. Let's just see if it's, let's go back. Also my little pointer. Yeah, here. Yeah. There it's coming, so that's it. Let's focus on that. See, it's hard to say. So I don't know the answer to that. Good question. But it's that conformational change that pulls the lagging foot forward. Okay, good.
Is there an AAT, or does it just have that functionality built in?
It has it built in. Yeah, it's not a separate nucleotide exchange factor. Good. All right. Covered most of this. So I have the steps written down for you. Now we need to talk about dynin. What if you want to go in the opposite direction? Fortunately, we're going to use that kinesin as our sort of poster child. This one's more complicated and less well known. It's the we've done that. Okay. So now, what if you want to go towards the nucleus? So you're going to need a dynin. All right, and it's ugly. All right, it's very complicated. So we're not going to worry about the actual walking mechanics. But the Dynon, the only thing that's sort of useful about this thing is Dynon has the motor. It's got the ATP binding domain, and it has the nucleotide binding domain. But then it uses this complex, which is called dynactin. So the dynactin complex attaches the motor to the cargo. And that's really all you have to know about it. It's got lots of different components. And notice it's got the whole mothership here. It's got spectrin, it's got anchorin, and that anchorin is useful for grabbing onto membrane cargoes. Right. So this, just think of, there's really two pieces here. There's the motor, which is called dynon, and then there's the cargo carrying capacity. In kinesin, the cargo carrying is light chains. Here, it's this ugly sort of dynactin complex. But that's as detailed as we're going to get about it. Okay, questions about that? So now we can move in both directions. We can actually move simultaneously on the same microtubule. That's a little bit easier for microtubules because there's so many cotofilaments. Lots of motors have access to different parts of that tube. Yes?
Why is this one so much more involved? Is this larger cargo?
No, because you can move large cargoes, in fact you have to, out to the plasma membrane. It's the way it evolved. You're sort of stuck with how you started. So, yeah. You can build, but it's hard to take. If you start messing with the machinery, then all kinds of problems happen. And we know that because you can mutate parts of that protein. Yes?
Can like the stress level of the cell cause the actin-myosin to move faster or slower? Or is it always at like one set speed?
It is one set speed because it's all set by the free energy released by one ATP being tweeted. Sorry. Yeah, and the stress level is sort of like heat shock proteins, or you get
like
osmotic stress or whatever. Yeah, that's detected by the signal transduction pathway. And then this thing doesn't really get into form. It says move cargoes, but just do it at your own pace. All right, so now let's do myosins. And the one we've been talking about is, this will be our poster child, is this myosin 2. This is the one that does a lot of stuff. It does muscle contraction, skeletal and smooth muscle. It does all this contraction. So that's going to be our focal point. Some of these actually, you notice they're pretty weird looking. Myosin 1 and myosin 5, I believe, actually bind to the plasma membrane. So they have little special duties. But this was the one that we focus on because it does a lot of the contraction function. So again, we talked about this. It has N-terminus is where the head domain is. It's got a neck or hinge region. It's kind of like kinesin does. It has a fibrous region. And notice there's no light chains at the end like there is in kinesin. Where are the light chains? They're around the neck region. They're up here. And these light chains, they're called myosin light chains. They get phosphorylated, they bind calcium, and so they're regulatory domains. And that's pretty much what all we're going to say about it. Okay, so you can actually do an experiment where you affix a myosin, say myosin 2, to a glass slide using lysine. You can coat the slide with lysine and stick a protein to it, like myosin 2. All right? And we're going to use this to figure out which way myosin is going to move this filament. Right? And so you can look at this in a microscope, and you can attach a fluorescent molecule either to the plus or the minus end of this. So that's a different way. You don't have to use the myosin decoration here because that's part of what you're looking at. So you don't want to use that technique. So you can mark this with a fluorescent label. And by telling which way the positive and negative ends move, you can tell which way the myosin is walking. Okay, so let's say we don't know that, we do know that it actually moves to the positive end. But let's say the positive end is, let's just figure that out. So if this thing is still, and so it's moving, so I need like here we go. So this is the black end is the positive end, and the negative end is this end, right? And so if I'm fixed to the slide and I'm a myosin 2, I'm still going to walk the way I'm supposed to walk. I'm going to walk that way. So I'm going to reach up to grab and pull, reach up to grab and pull. Which way is this filament going to move? To the negative end. You'll see the negative. So if the negative end moves, that means this is walking to the positive end because you're doing this, right? And that's what happens in muscle contraction. So you have a bundle of fibers. The positive end is blocked by a capping protein called CAPZ. Okay, so this is one end of the muscle fiber, contractile apparatus. And then there's another piece that's aimed in this direction. So you have a Z disc, it's called a Z disc. A Z disc here and a Z disc here. And then in between the actin filaments are these myosin filaments. But they're fixed. They can't move. The actin filament can pull the Z disc towards the middle of the sarcomere. So what happens is those myosin head groups reach up and grab a hold of the actin filament, and they fire ATP, we'll go through that, and then they walk. But what happens is the Z, they don't move because they're fixed. They pull the actin filament towards the middle of the sarcomere, and you get a muscle contraction. Because those sarcomeres are attached to each other in series, and if you pull, each one of those little pieces pulls in, what happens to your filament? Your muscle fiber shortens. That's contraction. Okay? So that's how you contract your muscle. It's because the myosin heads are reaching up and grabbing and pulling. And we know which way they move because that negative end moved that way. Right? So remember, here's the experiment. I'm the myosin. If I walk to the positive end, the negative end, it moves in the direction of the negative end. It seems counterintuitive. You think walking. So what if you cut my feet off and I was walking? The myosin would move, and you call this steady, it would walk that way. Right? But if my feet are attached to the substrate and can't move, then something's got to give. And so this thing is the mobile, the actin filament. So that's how muscle contraction works. Okay, so there's a contraction cycle with the mind. It's slightly different. Corner around here somewhere. Okay. Okay, so this is really wordy. So let's just look at this. All right. So we know that there's an actin filament that the myosin head's going to walk. There's the plus end. So we know this thing is going to try to reach up and move in this direction. It's going to go this way. This is not going to move. Why? Because it's attached to other myosin filaments, and they're stable. They're not moving. Think of, and I use this analogy, you have a bag of golf clubs. Okay, this is the Masters weekend, of course. So anyway, so you got a bag of golf clubs. The golf clubs are the myosin head groups poking out of your bag. It's a driver, and so those clubs stick out one end. What if you while Tiger Woods wasn't looking, you cut a hole in his other bag? First of all, he'd be really pissed off. But so you cut that in and you stuck his driver. Some of his drivers you took out and you stuck it in the back end of his bag, and it'd be really funny when you pick that up. But, okay, so you got clubs sticking out on both ends of the myosin thick filament, and what's happened is that's the thick filament that's going between the actin thin filaments. But those golf clubs are reaching up and grabbing both. This is a different actin filament than this one, and they're both walking that way and pulling the actin filament towards it. Right? So you got the golf club bag is the myosin thick filament, and it is thicker. It's much thicker. But it's reaching up with those drivers and grabbing and pulling it towards it. Okay, so back to this. So let's start at some point. It's a cycle, so you can start anywhere, right? So there's the actin filament. It is bound to the actin filament if and only if there's no ATP. So why do people go into rigor mortis when they die? After a few hours, they get very stiff. Why is that the case? What's one thing you can't do if you're dead? There's a lot of things. You can't go to golf tournaments or fool with Tiger Woods stuff. But you can't produce ATP. Is there some ATP even after you're dead? Yeah, it's still trying to do its thing. It's the brain that's dead, it's not you. Okay, but eventually you're going to deplete the cell's supply of ATP, And when that happens, you cannot unbind the golf club from the filament. So the myosin head is permanently attached to the filament if there's no ATP. So the role of ATP binding is to cause release. That's very different than the kinesin model. So this is different, but it's related, it's using ATP. But the binding of the ATP causes the myosin head to release. Okay, so that's the first function. It hasn't split, right? All right, so what it's going to do is it's going to then split that, and that's going to create some power, and that will actually cause the splitting of ATP causes it to cock forward. Okay, so that it goes like that. And so that's the walking motion of this. It cocks forward towards the positive end, because it always walks to the positive end. And what happened, though, is it didn't release the phosphate or the ADP. It held on to both pieces. Okay? And it's going to release them sequentially to do different things. Okay? So the next step is, so you're now cocked forward. You're now oriented. See, that's the binding site where it was, and now it's ready to reach up here and bind to that, because it's now reoriented towards the plus end, which is on this side. Okay, so now we're ready to go forward, and so we're ready there. What does it do? It's going to reach up and bind to that active binding site. So it's actually the actin filament, and that's the myosin binding site. That's what myosin binds to it. Okay. So that release of this phosphate, here it is, that triggers the power stroke. That release of phosphate causes it to go like this, pull towards itself. And that's what pulls the filament in the negative direction, like that. Okay. If this was unattached, it would walk that direction. Okay. Now, what causes the power stroke is the release of that phosphate. What does it do with the ADP? When it releases the ADP, it resets back to the original position. So ADP, kind of like in the multi-drug resistance protein, the phosphate release causes the loss of the substrate, and the ADP resets the ADPase. That's what happens here. You release the ADP, and now you're ready to go. You have no ADP or phosphate, so you must beware. You must be bound to the active filament, and you cannot release unless your body supplies you with ATP. So that's the whole cycle. But you notice it's very different from the kinesin deal. Okay, so let's construct a functional unit and make it a little bit simpler than what I was showing you with the golf bag. Okay, so this is a skeletal muscle fiber, and it's a functional syncytium. These are several multinucleated. Okay, and the contractile units are these proteins in there. Okay, that contain this structure. This is called the sarcomere, and let's look at this structure. So there's the Z disc. So that's the edge. That's the capping protein. Where is it bound? It's bound to the plus end of the actin filament. So that's the plus end, and that's the minus end. And if you can't have a sarcomere unless you have two pieces through it. You have to have a Z disc there and a Z disc there. And this orients these actin filaments in that direction. And so that's the plus end, and that's the minus end, right? And so this is the golf bag right there. And imagine little heads sticking out of there. In fact, you can see them here. See those little heads? There's the golf bag, and you've got golf clubs coming out of both ends. And that's so that it positions them close to the thin filament, because those little heads are completely uncoordinated. How is that different from kinesin? One head doesn't care what the other head's doing. That's the problem with a lot of people. But it's also the trouble with this. Okay, so they're reaching up there and grabbing, and as long as tropomyosin the blanket has been moved away, and that binding site, the myosin binding site on the actin filament is exposed, this thing will bind to it. Right? Okay, so they're all trying to reach up there and bind to the actin. And these are oriented in this direction. So what's going to happen is this is going to try to walk this direction. That one's going to try to walk that direction, but they can't go anywhere. They're stuck in the golf bag. So they're going to pull the filament, and remember in the experiment, if you walk to the positive end of my little stick, which way did the negative end go? Like that. That's the negative end, so it's going to go towards the middle. And this is going to go towards the middle. And what's going to happen to the two ends? They're going to come together, and you've got contraction. You've got shortening. Right? Okay, so that's what's going on here. That's the middle, and there's names for all this. I don't want you to memorize all the names. I want you to know that that's actin, that's a thin filament, that's a myosin, that's a heavy filament. That's really all you need to know. And then the Z-disc is the boundary of the sarcomere. So this is the functional unit of contraction, skeletal muscle and smooth muscle. Okay. Questions about this so far? All right. So there's a lot of proteins. I don't want you to memorize all these different names. Really, let me tell you the proteins that are important are the thin filament, that's actin filament, F-actin, so that's F-actin. The thick filament is a two-headed bundle of stable myosin 2. The CAP-Z protein is important because it's a stabilizer of the plus end, and it forms the Z-disc. Okay? But these other things, you can note them, but this thing is built to be very stable, so it's got these gigantic proteins like tropomodulin and nebulin and maybe Saturn or something. There are just all kinds of big proteins there to stabilize this thing. But we're not going to know that. You don't have to memorize all those components. Okay. Now, so we need to tell the rest of the story. How is this motion coordinated by calcium? And that kind of ties in with our signal transduction story. So let's do that. What happens is, let's get oriented here. This is outside the cell, right? Lumen of the extracellular space. So there's lots of calcium there, and you have a voltage-gated calcium channel, right? And what's going to happen is there's a neurotransmitter like acetylcholine is dumped on the skeletal muscle. Okay? So the skeletal muscle must have what kind of receptor? What's the best guess that you're going to, the quickest way to get a change in membrane potential is to choose what kind of cholinergic receptor, where the channel is actually built into the receptor itself. Remember the two flavors? Nicotinic, muscarinic? Why wouldn't you use a muscarinic? That's actually not the right answer. Okay. It's G-protein coupled, and the G-protein has to split, talk to itself, and all this other crap. All right, so you want to use the nicotinic because it is ion channel. It's a cation receptor. What happens is somewhere around here, acetylcholine has been dumped into the neuromuscular junction, and that nicotine, or not nicotine, but the acetylcholine has bound to the nicotinic receptor. It's open, and sodium preferentially moves into the cytoplasm when that happens. What is that going to do to the membrane potential as sodium pours in? It's going to depolarize, so it actually sets up an action potential. And it travels down this little region that's called the T-tubule. And it's called a T-tubule because it's like a sewer pipe. The plasma membrane goes in, and it goes down a tube because it's bringing that change in membrane potential deep into the muscle cell. Look at this thing. This thing is, it's got all kinds of sarcomeres. How are you going to get that change in membrane potential to go deep in here unless you make it go plunge into the cell? So you use these things called T-tubules. But they're they're little pipes made out of the plasma membrane. So when this voltage-gated calcium channel detects the nicotinic receptor doing its business, which is depolarized, this goes, aha, and opens. Because it's opened by depolarization. And it opens, and then calcium pours in. But what happens is, it's got this ryanodine receptor. It's not gated by IP3. It is a calcium-induced calcium release channel. So this calcium comes in here, binds to the ryanodine receptor, and you know what that does. Low concentrations open up the ryanodine receptor just like the IP3 receptor. It's the same thing. It's just not gated by IP3. So it's opened up. This is the muscle people call everything sarcose something. It's the endoplasmic reticulum. It's the smooth endoplasmic reticulum. This is a storage site for calcium. It pours into the cytoplasm. There's your calcium spike. And then immediately things try to get rid of it, right? But for a moment, you have this flash of calcium, and that's going to bind to troponin C, because that's the calcium binder. It's attached to tropomyosin, so it's going to pull on the rug, pretend that's a blanket. It's covering up what? It's covering up the myosin binding sites on actin. Myosin cannot bind to this filament because this is in the way. And so if you jerk it out of the way, using this complex that's decoded the calcium signal, it suddenly reveals a bunch of binding sites. And what are those stupid golf clubs? They're always looking for places to bind. And so if you give it a half a chance, it'll reach up and grab on to this and quickly go through the ATP hydrolysis cycle and start pulling. So this shows what the change in the tropomyosin. So it's blocking the myosin binding site, and calcium causes it to move out of the way, exposing the myosin binding site on the actin. So now those golf clubs can reach up and bind to this. And then very quickly, when the calcium signal goes away, this thing moves back into position and blocks that site. So if you go this direction, then it's now the blanket's in place. And this is a cross-section of the filament, and the tropomyosin is coming out of the page here, right? And the filament is going in and out of the page. And so this is a cross-section of, that's like slicing that and then looking at the end, right? So this is a cross-section of that filament coming at you. And so this side binder reassumes the position and it blocks that. So the golf clubs can't touch it anymore. Okay. All right, we covered a lot of stuff there. So any questions about this skeletal muscle contraction? Okay, before we go on to, we're going to briefly take a look at intermediate filaments because we won't do it today, but just a couple of other details about, we've talked about these things. Let's just, to give you, to complete the story, myosin 2 is not the only isoform. The only other two that we're going to look at is myosin 1 and 5, because they're the things that allow the myosin motor to bind to membrane cargos. And so myosin 1 binds membrane cargos. The reason it can do that is the tail domain contains phospholipids. Right? It binds phospholipids. It doesn't contain them, it binds them. And it contains an SH3 domain. What is that going to allow it to do? What type of protein will that allow myosin 1 to bind to? It's got SH3 domain. What's it looking for? Phosphotyrosine? Phosphotylinositol phosphorylate 3 position? No. Sarcomology 3. What protein has one that you know about? It's in the RGK pathway. It's in the MAPK pathway. It's the first thing that binds to the receptor when it becomes phosphorylated. RAB2. It's got an SH2 domain that allows it to touch the receptor. It's got an SH3 domain that's looking for a polyproline containing protein. So this allows myosin 2, because it has an SH3 domain, sorry, let me just jump down. That SH3 domain allows it to interact with proteins that have a polyproline motif. There we go. Okay. So it functions in all those fun things that we talked about: endocytosis, pinocytosis, testo microvilli. All right. So the last one is myosin 5, and that allows it to not grab the plasma membrane, but to hold on to vesicles. So you use myosin 5 to carry vesicular cargos. You use myosin 1 to actually link the actin cytoskeleton to the plasma membrane. So that's the difference. And then this is the workhorse that we were talking about that causes muscle contraction. Okay, that's it for you.